With us here, we have a very interesting enzyme, succinate dehydrogenase, a very important enzyme in the mitochondria of aerobic organisms with key roles in both Krebs cycle and the respiratory chain. But there's something interesting. If we lose activity of this enzyme, it all goes bad for humans because there's really not any metabolic bypass. So it can originate diseases such as Light syndrome here on the left and familiar pyroglomeia syndrome here on the right. So my name is Yaroslav Volkov, and we're going to briefly see the effects of this enzyme and how we can treat it. Let's begin with understanding metabolic diseases, which are a group of pathologies that can be acquired or can also be congenital and are characterized by disruption of metabolism or metabolic pathways affecting the body's ability to process and create various biomolecules. One of these biomolecules is succinate dehydrogenase, which is also known as mitochondrial complex 2. It is an enzyme involved in the Krebs cycle and the electron transport chain between the mitochondria. It's found within the inner mitochondrial membrane, and its function is to catalyze the oxidation of succinate to convert it into fumarate. And at the same time, it also reduces ubiquinone compared to ubiquinol, which is a molecule that is part of the electron transporter chain. This enzyme is divided in 4 subunits, A, B, C, and D. Now, we can also understand that it's a tetrameric enzyme composed of 2 hydrophobic units and 2 hydrophilic units on the bottom and the top, and it has 2 active sites, one for succinate and the other one for ubiquinone. When there's a deficiency of this enzyme, one of the possible diseases could be familiar pyroglomeal syndrome, which this one is a neuroendocrine tumors that are characterized by developing for the nervous system or any oxygen-sensitive tissue. So they can appear in the neck, they can appear in the head, the abdomen, near adrenal glands, pretty much anywhere in the body. These tumors are caused by mutations in 3 of these succinate dehydrogenase subunits, which is not really understood very well why they happen. Here's where we try to arrive to a possible solution, and there's a problem here because currently there are no specific therapies that target activity of this succinate dehydrogenase enzyme or genetic mutation. So treatment usually is only related to surgical removal of the tumors. And here we have a possibility using inhibitors for the enzyme, such as in anti-cancer therapies that has been researched lately in preclinical studies. And they have shown that using inhibitors induced tumor cell death, which is quite convenient because there's no more growth. And they precisely use the binding sites of ubiquinone and succinate. So these are the 2 binding sites they use. Another way of treatment is also with mutation correction. An example of this is using CRISPR-Cas techniques on the enzyme that a team performed by Bandera here in 2021. They precisely using CRISPR-Cas technique on these mutated cells of the enzyme, on particularly focusing this D subunit with HIK cells, and they obtained that the defects in mammal cells can be partially restored by correcting these mutations. So there's quite a good outcome in this possibility. Now, we will wrap this up with the commitment to sustainability, which many factors come into play. We can directly observe ODS-free, good health and well-being, because even though these syndromes related to the enzyme don't take many lives yearly, it's still a goal to reduce mortality ratios and also support the research and development of medicines for diseases. As we saw, these solutions using inhibitors or CRISPR techniques are bound to that. We can also think of ODST, reducing inequalities. It is also important in this situation as we cannot forget that research and development tends to be slow for these diseases that are not so common. So any intertwining with our diseases and research will be appropriate for developing each other's problems. And finally, ODS 12 for responsible consumption and production. Let's not forget that there's not a definite cause to the origin of these diseases. A possible one could be related to endocrine disruptors, EDCs, because we can find them in water, we can find them in food, in our clothes, they're pretty much anywhere, even in plastic. So a production of these products that consider avoiding the use of known EDCs could be a reasonable point to reducing possible causes for this disease. So it's a possible outcome we could take to solving this problem. With this, it will be pretty much everything, and thank you very much for your attention.